The first thing I'll note is, Marty, that was fantastic, and good to see Jack being memorialized like that. I will note that I was actually one of Dick Forbes's students, and my background in Calgary with archaeology goes back to 1980, so approximately 44 years. And for those of my friends who were in the audience and who were there at that time, when everybody else here says that's before I was born, it's true. So anyway, thank you very much, and thank you very much for this invitation. So what I want to do tonight is primarily talk about the dangers or some of the conceptual issues, let me put it that way, with archaeogenetic research, particularly ancient DNA research, which is what I'm most involved in now. I'm an administrator now, but I do this some of the time. And what I want to note first, and this is kind of my pathetic, I have done a lot of archaeology in various parts of Africa, most recently on a set of sites in northern Cameroon called the DGB sites, Good Bay, that Nick David at the University of Calgary and I have worked on at various points. And these are sites up in the Mandara Mountains, right by the border between Cameroon and Nigeria. And they're really amazing archaeological sites. There's about 16 of them in total. Stone-built architecture, rather mysterious in a variety of different ways. What a great sites from the point of view of having hidden passageways and stairways and all kinds of mysterious stuff going on with them. So they're a lot of fun to work with. These passageways were all hidden beforehand. Unfortunately, this image here, the one on the left, I took that from one of the DGB sites in about 2011, and at that point Boko Haram was already active in the furthest reaches of that, in the mountains on the slide on the left, across the border in Nigeria. So it became too difficult to work there after about 2014, and so I ended up writing a book about how Boko Haram, among other things, are understood in that area as slave raiders rather than terrorists. So that's just a note to, I do archaeology occasionally. So, but today I'm going to talk, this is going to be more like a lecture, folks, so I'm sorry, like a university lecture, because I can't do that anymore, so you're the subjects at this point. I'm not actually sorry, so here we go. So what I want to talk about is issues of how archaeology intersects with genetics. And I've been working on this over the last few years with David Reich at Harvard University, who has a lab that's devoted to the extraction and analysis of ancient DNA. And we've been working on a variety of different issues. I've gone from co-authored papers now to papers that have like 60, single-authored papers to papers that have like 60 co-authors on them. And you need a scorecard just to keep in mind who's doing what on some of these big projects. But this, for me, this came out of a paper I wrote about 24 years ago that was sort of in the prehistory of archaeology and genetic work. And that paper, Genes, Tribes, and African History, which among other things is the shortest title I've ever managed to give a paper. I've tried for 4 words since then, but I've never made it down to 4, and there's no colons. You can see in the, which are the bane of titles in archaeology. This was a paper about my frustrations with some of the interpretations, the way that genetic work was being used to interpret the African past at that point, because I found it overly simplistic and misleading. And so essentially I've been writing on and off on that ever since. And so this deals with aspects of that. But what I was talking about in that paper was very different kind of work than the work that I'm talking about now. And this is an image, this is the kind of image that you would see from that time period, from the early 2000s, as I said, back in the deep prehistory of archaeogenetic work. And what it shows, purports to show, is the spread of humans out of Africa and into other parts of the world based upon what are called lineage analysis, mitochondrial DNA, on the one hand for the maternal lineage and the non-recombining portion of the Y chromosome for the paternal lineages. And this shows then these very nice maps of haplogroups and their representation in various parts of the world. There's two things to note about these sort of analysis that were being done at that point. One of which is that these are lineage models. That is, they're models based on a single lineage of people who have inherited the same features through time. Your mitochondrial DNA is inherited from your mother's, etc., back to mitochondrial Eve. And similarly, the NRY is, if you're a male, is inherited from your father's, whatever. So they show a snapshot of a particular lineage of people inheriting genetic information over time. It's one thing. So, if you go back 10 generations, well, you've got one person back 10 generations that you're getting information about. Everybody else, all your other ancestors 10 generations back, their information's gone. The other thing is that they're based upon modern humans. That is, their DNA recovered from modern human beings. Do a cheek swab. This, all of these 23andMe, all of these tests now are done on modern human beings, and that is significant in a variety of different ways. As I said, number two there, the extrapolations of the modern genetic configurations to extrapolate to the history of ancient populations in the same area. Generally, this is the way it was done, and logically, under those circumstances, almost inevitably, they tended to involve assumptions of genetic continuity in an area. The further you push it back in time, if you're only looking at modern human DNA, the further you push it back in time, that assumption of genetic continuity gets to be a little tricky. So there were extraordinarily important and really useful ways of looking at the past. There's no question about that. But they were fundamentally limited in a lot of ways because you're taking DNA from modern people. It was also at that point a period of, let me call it genetic arrogance. Lucas Cavalli-Sforza, one of the people involved in this, used to say that this was going to replace archaeology. Because why do you need archaeology when you could just go and do cheek swabs? So that's not the way it worked out. So we get this. I think I put up as I gave to VV as an image. There's a lot of you can find these sorts of images almost ad infinitum. The ancient DNA revolution. Medieval DNA reveals African and Asian mixing along the Swahili coast. Ancient DNA. So what we've seen approximately over the last, depending on how you want to talk about it, within the last 15 to 20 years, has been a new revolution in the use of genetic material to interpret the human past. And that has been through the use of DNA extracted from ancient human remains. Okay. I have to qualify that a little bit because recently, over the last 4 to 5 years, you're starting to get the refinement of the techniques as such. I'm not going to talk about this now, but it involves its own ethical and scientific quandaries. The recovery of ancient human DNA from archaeological sediments. In cave sediments, for example. So the DNA not associated with any coherent human skeletal or other remains, but simply recovered from the sediments. You can imagine, as I said, the kind of ethical quandaries in terms of, for example, community control over genetic material that involves. But this use of ancient DNA has really revolutionized the way we think about the human past in all kinds of ways, and about the ways in which archaeology and genetics work together. Because remember, one thing, I'll just set this aside now in my a little bit of archaeological gloating, which is the geneticists realized really quickly, oh, in that case, we actually need the archaeologists to dig this stuff up. No more cheek swabs. We actually need them. And they became much more friendly at that point. 2 years ago, for example, Svante Pavot was awarded the Nobel Prize for his foundational work in the recovery of ancient DNA from human remains. I found out, I was down in McGill yesterday, and I found out that his father, through a complicated family relationship, was also a biochemist who received a Nobel Prize in biochemistry. So, which I had not realized before because family history. But you do not, I think, find very many cases in which a father and son in the same area receive successful, in the same area but doing completely different things, receive successive Nobel Prizes. So, what I want to talk about now is, I'm going to talk a little bit about what this involves first, and then about some of the problems with it. The first thing to note is that it involves a whole set of technological advances, and you'll see some of them up here. The selection of human remains, the petrous bone of the interior of the ear, for example, is particularly rich in DNA and preserves DNA quite well. Different ways. The main problem with the use of ancient DNA has always been contamination. And so ways to distinguish degraded ancient DNA from the contaminants in the environment, modern human DNA. This is one of the reasons why it's almost certainly always going to be easier to look at the ancient DNA of non-humans as opposed to humans, because if you're looking at bears and you have human DNA show up, it's pretty obvious that that's contaminated. And then a whole variety of techniques that I've talked about here that I'm not going to go into tonight at all, that I've covered under the rubric of next generation sequencing. Basically ways of handling, ancient DNA work is quintessentially big data, and ways to automate the analysis and recovery of degraded short strands of ancient DNA that have been highly damaged, and assemble them together computationally into a view of the genome of, or big parts of the genome of an ancient individual. So, it's not as if there's been one particular technological advantage that has just changed the world of ancient DNA research. These are big labs doing big science, and with lots of co-authors, as I said, and a lot of people working in tandem and spending a lot of money. One of the things that work did almost immediately was, and this is a pretty foundational issue. They begin to realize, and you can think of all of the, if you're old enough, you can think about all of the debates about migration versus stability of populations in different parts of the world in archaeology over the last 40 to 60 years. As I said, the use of modern DNA sort of tends to lead to assumptions of genetic continuity in areas. And what the initial work using ancient DNA indicated was that the genetic landscapes of different parts of the world looked very, very different over relatively short time scales compared to the way they look today. This was first done in Western Europe for a couple of reasons. One is because in a temperate environment, ancient DNA preserves better. This is one of the reasons, as an Africanist, one of the reasons that this ancient DNA work has only been really applied over the last 8 years approximately has been that just technically it's difficult to recover ancient DNA from hot, humid climates. So it became obvious very early on, and by that I mean in the late 2000s, that the genetic landscape of prehistoric Europe looked very different even 3,000 years ago than it does today. implying a lot of movement of people, a lot of population replacement of all different kinds. So that was an initial recognition from the very first work, and it's pretty foundationally important from the point of view of archaeology. And so you get maps like this from that time. An ancient DNA study links the corded ware culture of Northern Europe with the Yamna culture of the Eurasian steppes. Mass migration. Big lines on maps. I'm going to talk a lot about big lines on maps in this talk, because geneticists love big lines on big maps. So this shows a good set. It doesn't come up as well, actually. There's a very light green color back there that's supposed to show the continental areas, and it doesn't seem to be translating. But you generally get the area I'm talking about, I hope. And you can see the lines anyway. So lots and lots of work in the late 2000s and through the 2010s. David, in his book, Who We Are and How We Got There, at least does not use the word mysterious. I hate that word so much. But you notice that everybody else here is using mysterious. Remember, everything archaeological has to be mysterious. There's a rule. I don't make these things up. Key mysteries of ancient human societies. So ancient DNA is going to solve, because us archaeologists are too dumb to do it ourselves. It's going to be ancient DNA that does it, because it's big science. It's not all this boring digging stuff up. There's problems with that, though. And this is, I love this map, the hype cycle of ancient DNA, which is totally true. But I don't want to be too hard on it. And the innovation trigger, the idea is sort of around 2005, 2010 that it might be possible to recover dinosaur DNA. This stuff, we have a lot, though we can blame these Jurassic Park movies on this, remember all that? Actually, it was a little bit earlier than that, but you get it. The peak of inflated expectations, the trough of disillusionment. Svante Pavot, I don't know if you remember, but the first work on this was the recovery of Neanderthal DNA. The first real impacts of this involved the recovery and sequencing of Neanderthal DNA. The first time Svante did this, he got it wrong and had to quite embarrassingly, retract the first study because the DNA was contaminated with modern human DNA. Totally reasonable sort of thing. He handled it very well. But so this picture of the Neanderthal in the National Geographic indicates that there was contamination is a big problem. And then he went back and refined his procedures and did it right. So this between about 2003 and I think the corrected work was at about 2008. So it's a problem. It's this is extraordinary work. And I have, despite my periodic bitchiness about being an archaeologist with these folks, that this is something that I have a great deal of respect for these folks and the work that they do. And then the plateau of productivity. But the one thing I'll note is if you go back and look at it, carbon-14 dating went through exactly this same cycle. So it's quite reasonable, if we think about radiocarbon dating now as a relatively well-tamed technology, so to speak. But in the late 50s, early 60s, radiocarbon dating was the Wild West in terms of interpretation, before calibration came in particularly. So this is not peculiar to ancient DNA work at all. I want to talk now about some of the challenges of interpreting ancient DNA work, and this is what I'm going to talk about quite a bit today. Just because I think it's kind of an interesting thing from the point of view of thinking about epistemology and the way we understand knowledge and how this all works. So, another, like Dick Forbes, another giant of the Department of Archaeology at Calgary, Nick David, if you ever took world prehistory with Nick David, then you knew how the world worked in prehistory. He was pretty emphatic about that. So you take these classes, I think that was actually probably Archaeology 102 at that point instead of 101. But the idea that you understand the past, particularly if you're not a specialist, if you're a geneticist, you've only ever taken Archaeology 102. You've never taken anything beyond that. And so you interpret the past through what you know. So I call this historical saliency. What are the things you know about the past? A limited number of events, of processes that are named, not necessarily well understood, but named, that you, and this has been a problem with genetic research, archaeogenetic work for the last 25 years. What do you think, when you find patterns in genetic data, what historical processes do you connect them with? Okay. One of the things that genetic data is still not very good at is giving you time depth, giving you dates for things from the genetic data itself. Still difficult to do that. So how do you date this stuff? So in African archaeology, for example, everything's the Bantu expansion. Everything in genetics is the Bantu expansion in Africa. That's kind of mean, but not as mean as you might think. This is the expansion by which a family of closely related languages called the Bantu languages spread over much of equatorial and southern Africa over about the last 3,000 years. Complicated process, the degree to which involves human movement and population spread and language spread is still something that people debate a lot. There's a lot of data on this. But the number of cases, basically anything that happened vaguely in the Holocene and late Pleistocene in Africa south of the equator is going to be ascribed by geneticists to the Bantu expansion. Because it's a thing. And all of your Archaeology 101 texts are going to mention it. In the same way, the spread of Islam. Okay? It's another big event. People move around. When people move around, obviously there's got to be genetic consequences. Okay? So if you got something that looks vaguely, if it's an east to west move along the North African coast, well, it's got to be Islam. And I'm not really exaggerating here, folks, that this question of historical saliency, understanding the past through reference to events that you've heard about before, is a real problem. Or, for example, things like the trans-Saharan slave trade. We find evidence of genetic motifs from sub-Saharan Africa in northern Africa and indeed in southern Europe. We definitely find them there. They are reflexively associated with the trans-Saharan slave trade. And then you find that some of them, when you start doing ancient DNA research, you find that some of them are showing up in the late Pleistocene, and all of a sudden that gets a lot more complicated, because the trans-Saharan slave trade did not exist in the late Pleistocene. So these sort of ascriptions of historical interpretation to genetic patterning is actually a major problem. Okay. Limited number of well-known events. Genetic processes, as I said, uncertain spatial and chronological ranges. You can tell that I'm a professor. So the identification of the two then, the genetic pattern and these historical events, becomes almost inevitable in some cases. I want to give you an example of that, and this is a really pernicious example because it involves the first really well-known ancient DNA research to be undertaken in sub-Saharan Africa. And as I said, it's challenging to do it in sub-Saharan Africa because tropical environments, essentially. So it's not actually the first that was done. The first ancient DNA research done in sub-Saharan Africa involved the recovery of ancient human DNA from tooth calculus from a jawbone found in Ghana, but that's never really gotten the attention that this has. This is this paper, Ancient Ethiopian Genome Reveals Extensive Eurasian Admixture Throughout the African Continent. And it was, so this is from Ethiopia. Now the reason that it's actually quite predictable that ancient DNA from Africa is first going to come from the Ethiopian plateau, because it tends to be drier and cooler than much of the rest of sub-Saharan Africa. So it makes sense that it's going to come from, this is from Mota Cave, and this individual is sometimes called Mota. So a skeleton recovered from this cave, and ancient DNA was recovered in turn from the skeleton. And this paper was then written. And it got a lot of attention. And it got a lot of attention in this way. First ancient African genome reveals vast Eurasian migration. Because the interpretation undertaken in this paper was that farmers, Neolithic farmers from the Near East had spread throughout sub-Saharan Africa in the late Holocene and contributed large, significant proportions of their DNA to African populations in sub-Saharan Africa across the continent. Out of Africa and back, ancient, the term vast comes up a lot. Now also, the archaeologists working, this was about 2014, the archaeologists working in the area had some problems with this. And the problems are just straightforward archaeological problems. Eurasian farmers coming in from the Near East at this point would have been wheat farmers. We know what their domesticates were really well. And if you take a look at the modern distribution of wheat in sub-Saharan Africa, you see this. And you see a lot of wheat in the Ethiopian plateau. And the reason for that is that wheat is a winter rainfall, and also in southern Africa for the same reason. Wheat's a winter rainfall crop. It doesn't do really well in the summer rainfall regimes of tropical Africa, which is why you find crops like sorghum and millet and non-cereals being used in this area. So raises the question, how are Eurasian farmers going to spread across much of sub-Saharan Africa when their crop systems are incompatible with the environments in these areas 3,000 to 4,000 years ago? This goes back to a very old idea in African history, sometimes called the Hamitic hypothesis. The idea that civilization was always being brought to Africa from outside the continent. This is the best known work, probably the prototypical work, originally from the 1930s, C.G. Seligman's The Races of Africa. And the civilizations of Africa are the civilizations of the Hamites. Light-skinned invaders coming from outside the continent, bringing light and civilization to the benighted Africans. So essentially what these news writers were doing from 2014, 2015, it was like the Hamitic hypothesis had come back again. Well, of course Eurasian farmers come in and spread across Africa and replace African farmers. And we've got a problem. Correction. It turned out the coding that had been used to interpret the genetic, there were lines in the computer code that were wrong. And so they had vastly overestimated, and this was like, the editors at Science, believe me, did not want to hear about any objections from any like dirty little archaeologists talking about this. This was big science, remember. But then one of David Reich's students, Pontus Skoglund, found that this team had actually miscoded the data, and they had to retract the paper rather embarrassingly. And it turned out that fundamentally there were a lot of Eurasian farmers on the Ethiopian plateau, which we always knew. They're speaking Semitic languages. I mean, it's pretty obvious that there was a lot of movement back and forth between the Arabian Peninsula and the Ethiopian highlands. That wasn't any surprise. So they had to walk back the rest of it. The original claim, 4 to 7% of most African genomes originated from Europe, from these European farmers. That's a lot. So this was an embarrassing problem. And it was this question of historical saliency again. A lot of people make the assumption that if cultural advances happen in sub-Saharan Africa, they have to be brought in from somewhere else. It's a bad problem. It's a bad problem to have in 2014, 2015. Other issues, scales of analysis. And this is a different kind of problem. And I put it down here at the bottom. Geneticists tend to do forests, archaeologists tend to do trees. The scales of analysis, because archaeogenetic research is prototypically big data, it's also expensive. And so the successful archaeogenetic labs are those labs that are going to build big conclusions. And big conclusions tend to be continental scale conclusions. People moving from place X to place Y continentally, that kind of scale. And it's very difficult from that point of view because it tends to be a different scale than archaeologists work on. We tend to work on smaller scales, tend to work on more fine-grained analysis. We often make big conclusions, but they tend to be built up over different time scales. So this, this idea of large geographical and temporal scales, relatively discrete population movements, that is big migrations, rather than small scale, long-term movements of people, contacts between people. As I said, I've showed you this map before, and it's interesting because that light green color, which shows the continental margins, actually comes way better here than on there. But there you've got Western Europe, you've got Sub-Saharan Africa, you've got the Black Sea and so on. This is quintessentially an archaeogenetic map. Big population movements. This probably did happen. You know, this idea that you do get populations from West Central Asia moving into Western Europe, it almost certainly did happen. But it's presented as a big, discrete population movement. And it almost certainly did not happen that way. When you take a look at, when you take a look at the actual data itself, you tend, I'm going to go into one of these maps here in a second, one of these structure diagrams in an African case here in a second. But you start to see a lot more messiness in the actual individual genetic data than is often the case in the conclusions. And we find the same thing in Africa as well. Big maps of the continent with big arrows drawn on it. And the question then becomes, how realistic are those arrows as anything but imagined portrayals of population movements? We know the Bantu expansion happened in some form, but for example, this movement that's located there, 3,000 to 1,000 BP, going east to west, just on the southern extensions of the Sahara, that's actually a much less well-defined movement. In fact, it's difficult to know what that means at all, but it's just another colored line on a map. There are geneticists who recognize this question, I will note. Krishna Virma, who's one of the folks that I work with periodically, is someone who's really recognized that in order to understand what this means in real life, that geneticists need to be paying more attention to small-scale population interactions. The idea that you can understand in some fruitful way continental population interactions without having any idea whatsoever what this means on a local scale seems to a variety of people, including me, fundamentally misinformed. So Krishna here is talking about looking at individual communities. And the interesting thing about this is, although there's lots of analysis, for example, in Sub-Saharan Africa, or in Africa as a whole, going on at a continental scale, there's almost no analysis that looks at, for example, genetic interchange across ethnic boundaries on a local scale. Almost none. Very, very little evidence of how that actually happens. What you do is you take scattered samples from different parts of Africa, you subject them to ever more complex analysis, and you make continental conclusions based on that. And this closely ties to the third set of problems I have, which is the fact that human populations are complicated. And sampling of genetic material from modern human populations or from ancient populations is also complicated. And this is something that's often underestimated in archaeogenetic work. Sorry. And I'm going to talk both about, in this case, about ancient DNA and about modern research, because the problems are parallel in the two cases, both using DNA from ancient individuals and using DNA from modern populations. Because one of the best examples I have is from using DNA from modern populations. One of the questions is simply, how do we understand population structures? And part of the reason I talk about this, as you'll see in a minute, is because working in northern Cameroon, up in the Mandara Mountains, I spent a lot of time with Ian Robertson and with a variety of other people working in areas with really complicated population structures. We're trying to figure out where individuals came from and who they might be marrying and who their parents might have married was actually really quite a complex question. And so we expect that ancient populations are going to be complicated in the way that modern populations are. So the fact that you have a skeleton from location X means that you may be able to make continent-wide conclusions in some ways about what's going on genetically in prehistoric Africa, but it doesn't necessarily rule out the idea that sampling somebody from 10 kilometers away would have given you a very different picture. So let me give you an example of this. So there's one of the, unfortunately I have to deal with these periodically. This is called a structure diagram. And fundamentally it's a diagram using a set of assumptions about the genetic characteristics of a set of individuals from different places in the world. In this case, we have Britain, we have the continental United States, Germany, a variety of areas of Sweden, a variety of areas in Norway and Russia. And so each of these vertical lines is an individual. And each of these individuals has a different set of genetic characteristics. And if you do an analysis of the relationships of these individuals within and between populations, you tend to get you get these different distributions of relationships of individuals with different genetic characteristics. The number of the K numbers is the size of the source population. So the assumption is here you've got a variety of populations, all of which have contributed to the genetic ancestry of sampled individuals. And each of these vertical lines is a sampled individual. So you make a set of assumptions. Well, if the number of source populations is 2, you get the top diagram. You only get 2 colors on it, 2 source populations. If it's 3, second diagram down. If it's 4, the third diagram down. If it's 5, the bottom diagram. And those numbers are arbitrary because we don't know in these cases, even with modern genetic samples, we don't know how many source populations actually contributed to the characteristics of modern people. So this sort of structure diagram there then is a useful way of looking at the genetic variability of populations. But I want to show you now, so I want to show you a different example. And as I said, this is undertaken using modern genetic material, but it's modern populations that I know in Africa. This is from Sarah Tishkoff, an article done in 2009, The Genetic Structure and History of Africans and African Americans. And what I want to look at, you can't see it here unfortunately, but here we have K2 through K14, so between 2 and 14 modeled source populations. We have individuals from a whole variety of African ethnic groups arranged vertically down at the bottom, and it's purporting to show the relationship between them. So what I want to do is just zoom in. You still can't tell here, I'll zoom in a little bit more, on a particular area here. And these populations that I've circled are the ones that I'm most interested in. So if I go back, we go from this to that. And the reason I'm concentrating on these populations, that these are the populations in the Mandara Mountains that I know very well. Populations like the Mara, the Oldame, Giziga, Mandara themselves, Kodako, Zulgo, Poroko. This is where I did my PhD. And so while I was a student here at the University of Calgary. So you can't see the whole expanse of modeled variability there, but fundamentally, these populations look as if they're pretty heavily related to Afro-Asiatic populations in different parts of Africa. Now I want to just jump into what these maps look pretty clean, okay? Certain amount of relationship between the populations. For most of this area, it's probably K2 to K5 is going to give you a lot of the variability that you're actually going to see. Well, this shows where most of these populations are. This is where I work in northern, where I did my PhD in northern Cameroon, the area particularly right there. And so you see a lot of those same names: Mata, Uldame, Podokwo, Orza, Wandala is the same as Mandara, and so on. This is populations there are the same ones as in this genetic chart. Beautiful area of the world, really interesting people, very, very nice people. But what I want to note here now is those are the ethnic groups involved, and this is the way that Sarah Tishkoff collected data on this region. If you actually spend any time in this region, this is the next one's the same map. That shows the number of self-identified groups that people belong to. You go from this to that. And you find out this is a whole variety of different groups of populations in there that identify themselves as separate groups, but that are not represented by the tribal names that the geneticists were using. It gets worse, because then you start to think about what migration looks like. And what you see is just a huge amount of people moving around in this region. People are just moving all over the place all the time. So these are, again, colored lines on maps, but the difference here is that the maps themselves are much, much smaller. This is showing people moving over distances of 10, 20 kilometers. And we have just huge amounts of evidence for migration of this sort. This is the area around the DGB sites, those sites that I was showing there a few minutes ago, where I'm working now, and even in there you've got lots of evidence of migration. So that I did my PhD primarily with a group of people called the Placha in northern Cameroon in the northeast Mandara Mountains. There are 750 Placha. Two subgroups, one of them called Placha Kapa and Placha Dunglalai, the other one. One of them had about 500, one of them has about 500 people, the other has about 250. And even within those groups, there are communities coming from different places. What that means is it really depends on not only sampling a population group, but precisely where you sample may give you quite different population affiliations genetically. People may call themselves the same thing, but they're coming from quite different places. Just as you might find exactly the same thing in this room. There is a tendency to think that population structure and population interaction in other parts of the world is not quite as complicated as it is, for example, in Western countries. But in fact, it is. And so these questions of population structure, one of the problems with these structure maps, bouncing back and forth, one of the problems with these maps is that they make no distinction, they make no provision for distinction of populations between the individual and the ethnic group. You're an individual who is a member of an ethnic group, and there's no way of thinking about any kind of intermediate level of structuring. And that's a major, major problem, because that's not actually the way that human populations work. I told you I was going to lecture you. And so we have this set of problems that's really heavily associated with this. Deciding the numbers of populations, an arbitrary, largely arbitrary question. You go with K2 or K6. Difficulties for accounting for recent admixture, availability sampling, and so on. So this is, again, a set of problems. Part of these are intrinsic to doing the DNA work. Part of them are just the relationships between ancient DNA research and archaeology. None of them are fatal problems, but they're all things that have to be taken into account. I just want to know one thing really quickly. So this is a paper I worked on with David Wright's team at Harvard, involving the recovery of ancient DNA from some skeletons in a site called Shumlaka in northwestern Cameroon. A couple of skeletons dating to about 8,000 years ago, and a couple more dating to about 6,000 years ago. And in this case, probably related people coming from related populations, in all these cases, relatively small population in this area. But one of the interesting things about this research is that it throws light on all kinds of different, not merely local population affinities, but because we all inherit, if we're not looking at lineage-based models anymore, but looking at the whole genome, we all inherit an immense amount of genetic knowledge, of genetic information from all of our ancestors. And this allows us to make, draw conclusions about population affinities at much larger scales. It does allow us to do that. There's no question about this. And so we were able, using those data from 4 individuals, to start making connections between those 4 individuals and individuals all over West and Central Africa, including ghost populations. And these are populations that existed in the past, that have passed down their genetic material, but which do not exist anymore. So they've either disappeared or they've combined to make modern populations. Some of these are, as we see with modern humans, particularly in Europe and Asia, some of them are from archaic populations. For people with primarily European and Asian ancestry, this might be Neanderthals or Denisovans or other archaics. We have the same thing going on in Africa. So these, and the techniques are extraordinarily rich. The things that we can do with these techniques now are really quite amazing. And they are going to revolutionize how archaeology works. But they need to be approached with a good deal of care. They need to be approached with, and with some humility, which is something that, and a lot of conversations between archaeologists, geneticists, and other researchers. Well, that was it. Okay, I thought I'd scrubbed all the. 因为我是昆山杜克大学的副校长，我会说谢谢大家，如果你有问题，now's the time for questions. Sorry, and my Mandarin is terrible.